Hello, this is Mrs. Kohler, and this is your last podcast of the DNA unit. Today we're going to talk about mutations. Now, mutations are things that you normally hear of as something bad, that they usually cause something to go wrong, and normally that is the case. Normally mutations are not beneficial. But we're going to talk about 3 different types of mutations, and there are some cases where mutations can either be a good thing or they don't even affect the organism at all. So we'll talk about the 3 main types and how they do or do not affect the way that you turn out. So, definition of a mutation is any change in the nucleotide sequence of DNA. So anything that changes from the normal way your DNA is supposed to be is a mutation. And the 3 main types that we're going to talk about are substitution, insertion, and deletion. So the first type of mutation is a substitution mutation. And a substitute just means something's there in place of what should be there. So in this case, where there should have been an a G was inserted instead. So one base just got substituted for another base. Now, in some cases, that substitution doesn't actually cause the protein that's produced by it to change. Remember when you were doing your codons, sometimes more than one sequence codes for the same codon. For instance, leucine has 6 different codons that can code for it. So if the letter changes to something else, but it still codes for the same amino acid, it's not actually going to change the genes in the individual. It's still going to produce the correct protein. And that's when it's called a silent mutation, because there has been a change from the original, but it's not doing anything to the organism one way or another. So there's 2 things that could happen in a substitution mutation. In this first case, GGC, which is codes for glycine, turns into AGC, which codes for serine. That would be a change in the protein because you're substituting serine for glycine. On the other hand, if you start out with CUU, which codes for leucine, and it changes to CUC, which also codes for leucine, the protein hasn't changed at all. So a substitution can either change the protein or not cause the protein to change at all. It can do one or the other. Now, the second type of mutation is called insertion. You insert something, you add something. So in this case, an extra base or bases are added somewhere into the DNA sequence. So in this case, an extra TGG gets added partway through. Now this is almost always going to cause the protein to change. Third type of mutation is called a deletion. When you delete something, you take something out. So in this case, the G and the G in the middle got deleted, so that now you have a shorter sequence. That also almost always causes the protein to change. So insertion and deletion are what are called frameshift mutations. And what that means is as the tRNA goes down, it reads the mRNA in groups of 3. But if you insert something or delete something, if it's just 1 or 2 of them, it's going to throw off all of the codons from there on. Because if you insert or delete just one thing and you're going by groups of 3, every single thing after that is going to be changed. And that's called a frameshift mutation. So insertion and deletion are much more serious mutations than substitution is. So when you're talking about different types of mutations, substitution sometimes causes a change, sometimes doesn't. Insertion and deletion almost always do. Now, you're going to go on to the practice in your packet, and you're actually going to practice doing some mutations, and you're going to say whether they're insertion, deletion, or substitution, and whether or not they change the protein. In addition, you're going to want to pay attention to this because 1/2 of your test is going to be the multiple choice, but the other half of your test is actually going to be creating a DNA sequence and then mutating it and creating one of each of the 3 types of mutations and then showing whether or not it changes the protein. So this is something you want to make sure that you know how to do. Have a great day, and I'll see you in class.
There's one more type of mutation that I want to make sure to point out, and that's called a nonsense mutation. A nonsense mutation happens when the codon that was supposed to be there gets changed to a stop codon instead. So then instead of finishing the protein, the cell just simply stops making the protein altogether. Making a stop codon is a serious mutation that can cause a protein to not be able to be functioning in a living thing. So nonsense mutations are something that you should watch out for when you're doing your end of the unit assessment.